Hello, everybody, and a very warm welcome to everyone attending our webinar today. My name is Claire Horsgood. I'm one of the VPs here at Mac Clinical Research. I'm very pleased to be hosting this webinar for you all and really grateful that you're able to spend some time with us today. So without further ado, we will look at our speakers for today. So there's Dr. John Connell. John is our Chief Scientific Officer here at Mac Clinical Research. You can read about his credentials in front of you. We're also joined by Professor George Dorfner, who is the Head of Clinical Trials at the Siesta Group. Thank you for joining us today, George. Both of whom are industry leaders. We're very, very pleased to have them with us today. And I'm going to be handing over now to John Connell.
Thank you very much, Claire. Hello, everybody. Thanks for joining. I hope this is going to be a really informative and helpful session on the use of EEG biomarkers in clinical research. We've got some time at the end for discussion and questions, so file them in when you can do. In the initial part of this webinar, I'm going to be summarizing the way in which a full-service CRO like Mac Clinical Research is using EEG today to support clinical studies really across all our phases of drug development, both at our own sites and also when we're partnering with sites around the world on larger later phase studies. Next slide, please. What I'd like to touch on first is why we bother really. Unlocking the brain. Why is EEG so important in clinical research? I'll take a nostalgic step back in time at the moment and picture a scene in, I think it was 1986 when I actually ran my first EEG. I was in a small clinic in Manchester. I had a very, very large IBM AT computer that almost filled the room on its own. Lots and lots of amplifiers and about 4 or 5 electrodes attached to this poor young individual where I was testing the effects of Ritalin. I'm going to come back to that image a bit later on because I want to show really how we've moved forward in the last 40 years and how that's, EEG technology is now initially still giving you the great same scientific data that we've always been looking for, but now is really much more usable and user-friendly, and data is much more available and higher quality and so much more quickly as well. Okay, EEG is really an invaluable tool that provides an insight into brain activity in real time. It's something that allows us to assess brain function, monitor safety during clinical trials, and can be pivotal in supporting drug development. It's really the backbone of many CNS studies these days. Throughout this webinar, we're going to explore how EEG is helping us accelerate particularly early development, but also safety assessments in later development as well, by detecting abnormalities in brain activity and providing deeper insights into the effects of experimental treatments. We're also going to highlight some of our expertise, including the dedicated EEG team we have working on studies within MAC, but also the work we've been doing more recently with psychedelics and also in our sleep laboratories, all designed to ensure that we can provide the highest standards for every study that we undertake. Okay, next slide, please. I'm going to give you a few slides really to let you know exactly who MAC are, because you actually may not have heard of us at all. Science is at the heart of what we do. We are a full-service CRO. Over the last, I suppose, 5 years or so, we've worked in over 50 therapeutic indications, many sponsors and many clinical trials. Particularly over the CNS ones, it's amazing how much we've been doing with EEG and increasingly are using EEG in these studies. In terms of physical sites, we have 10 sites across the UK. If you go on to the next slide, actually, it'll show you where they are geographically located. Across the north of England, and this supports really clinical development all the way from first administrations in man in our Manchester facility to the end of phase 4, really, in many of the later phase sites where we run bigger patient trials, both within MAC, but also we collaborate with centres throughout the world. And we're referring back to how these centres are very important and how each of them is using EEG in a slightly different way as we go through this very short introduction to MAC. Next slide, please. So we know what MAC is. Why bother with EEG then? Back to the science. EEG is, at its most simple and non-invasive method for recording the electrical activity of the brain. The brain generates electrical signals, brain waves. And what EEG is, it's a gold standard way of capturing these brain waves and turning them into something which is measurable and perceivable. It's done by electrodes which are placed on the scalp, They pick up the electrical impulses produced by the brain cells and provide us, as I said, a very direct and very real-time reading of what is going on in the brain. What makes EEG particularly valuable for all our stages is that they can be used across different states, whether the subject is awake, sleeping, under sedation, or as we shall talk about later, they're actually in a changed state of consciousness due to a psychedelic experience. In all these situations, EEG provides that clear, continuous record. Real-time data from EEG is particularly useful with both qualitative analysis, where we interpret visual data such as frequency and amplitude of brain waves, but also quantitative EEG, QEEG, where the data is analyzed numerically, detecting different patterns. And I know that Professor Dorfman later on will be really touching on this in a lot more detail, how these biomarkers are there and how we use them in particular patient areas. But my focus is really going to be on the breadth of what we can do across the phases of development. The main reason why I think it's being used so much at the moment is it's a very cost-effective way of looking at brain activity. It offers a wealth of information, and so we'll give illustrations in a short while. Some of the studies we're doing have to be done anyway. Measuring EEG is just the added icing on the cake, if you like, where you're actually getting measures of cognitive function you can correlate with other behavioral changes. Next slide, please. Starting off really with EEG in early development. The picture here is of our phase 1 unit in Manchester, accredited by the MHRA for all trials of clinical trial. And what goes on here, I suppose, is drug going into man for the first time. These are pivotal studies where you're looking for safety, tolerability, and PK of brand new chemical agents. And in early development, EEG can really play a pivotal role. By monitoring brain waves and activity in real time, we're able to understand how the drug is affecting the brain. But why is that particularly important in these early phase studies, like single ascending dose studies, where drug is going into man for the first time? The main reason is that in these studies, you'll quite often be working with people who are very healthy, but also doses of a drug you're never going to see again. So putting in measures of electrical activity of the brain, what is going on as the drug is in the body, gives you the opportunity to really look at high concentrations of a drug that you may never see in the patient trials that follow. At a very simple level as well, it can be useful in showing how the drug will cross the blood-brain barrier. The beauty of it is that this is going on alongside the standard measures. In an early phase study, you're looking for safety, PK, and tolerability. They're still going on, they're not affected, but we're able to measure EEG in these studies to see what is happening. It's also very important in confirming target engagement, providing direct evidence that a drug is actually interacting with the brain region or a network, or even simply getting into the brain. Animal data is there, but it is uncertain. This is the first time you have to actually show that you are actually getting brain penetration that can really help with making go, no-go decisions at an early stage. More importantly, it allows for the rapid iteration of drug developments. By monitoring EEG early in a trial, researchers can determine if a compound is effective or whether it should actually development be discontinued. This can really contribute significantly to development timelines. Next slide, please. So really in EEG, there's two things in early phases, two things we're doing a lot of. We have some people having electrodes applied here on the side, good old-fashioned way of gluing electrodes on at the top there for very small studies where you're looking at perhaps a very limited number of electrodes to more complex 64, 32 channel EEGs using CAP systems. We're doing pharmaco-EEG regularly, and this will be talked about a lot more later. Clinical EEGs, though, are also quite important, and we can just use these to ascertain that people are healthy when they take part in clinical studies. Next slide, please. Moving from the healthy volunteer studies into the patients, we do, as I mentioned, regular, normal clinical EEGs. These are not a true biomarker per se. They can still be very pivotal in making developmental decisions as you're working through a drug. Animal data perhaps has shown that the drug which is being developed has got proconvulsant activities. We've been asked on a number of occasions to do EEGs before and well after a treatment to show that the treatment has not had any effect at all on changing the way the brain is behaving. May have seen changes in animals, but how is it going to be in humans? This is a very early way of telling. The other thing we can do is use it for screening to make sure we only get people onto studies who are appropriate. So for a drug where there is perhaps a danger of inducing seizures, we will look before the study starts for epileptic form activity, and we can then sort of get these people not into studies to help them from a safety aspect to really protect the study. So through continuous EEG monitoring, we're allowed to detect abnormal brain activity, and that can be quite important, I think, really across all the phases of drug development. These data are also very important in supporting regulatory submissions by providing direct evidence of the drug's safety profile. Giving detailed value of the brain's response to a drug is something which I think is very important when you're trying to pull together a full safety package on a drug which is particularly CNS active and which may have had some data in animals which you have some concerns about. Next slide, please. Most recently, I think, where we've been looking at EEG is a fascinating area of psychedelic research. This is an area which I saw 10 years ago I had no idea we'd be doing so much of. But the rise of psychedelics in treating many CNS disorders or being suggested for treatment of CNS disorders has led to a whole raft of research that's ongoing at the moment. At MAC, we've done over 15 studies using a wide range of psychedelic agents, psilocybin, LSD, DMT, ketamine. One of the things that they've been doing really, we've been looking for is under very safe, very controlled conditions to have people have psychedelic experiences. The setting is very essential for these, and we're very experienced at how we can handle these sorts of studies. But one of the questions we're often being asked is, great that you can do that, great that we can have these people experience a psychedelic experience, but what is actually going on in the brain while it's happening? Can you do EEG measurements during some of these very high doses of drug like ibogaine or DMT? And the answer is yes, we have done, and yes, we have been doing that regularly now on a number of studies. Next slide, please. Psychedelics are interesting. What we're looking at is the effects of psychedelics where they can be profound during altered consciousness and perception. These ways are not easy to quantify through subjective measures alone. That's where EEG offers a distinct advantage, providing objective data that can really help us understand the underlying neuronal mechanisms at play during psychedelic experience. It allows researchers to track changes in brain pattern as individuals undergo their trip, if you like. It may be something which lasts for 20 minutes, it may be something which lasts for up to 8 hours. But what we're able to do is observe phenomena like alpha desynchronization, where the usual rhythmic oscillations of the brain are seen to slow. We're also able to look at analyzing different regions of the brain and how they interact while the trip is ongoing. Through this research, EEG has a potential to help identify therapeutic signatures during psychedelic states, eventually mapping the brain activity that correlates with beneficial therapeutic outcomes. Very promising area for mental health treatment, particularly with conditions like depression, PTSD, anxiety, where psychedelics have been tested as novel therapies. It clearly relies on a very strong relationship between the scientific team working on these projects, the clinicians, the staff really setting up the correct environment so you can control the psychedelic experience. It's actually a fascinating scientific experiment to be conducting and something which we're now doing on a regular basis. Next slide, please. Very briefly touch on the fact that we do also look at sleep. We have a sleep laboratory. It's our Liverpool facility. It's a 5-bedded laboratory with full overnight PSG capabilities. Next slide. Although we're able to look at a full range of sleep assessments, this could actually take a webinar on its own, so I'm not going to deal with that here. What I will say, though, is we are being increasingly used when developing drugs which are CNS active to look at things like sleep assessments, to look at changes in REM, non-REM sleep, because quite often how an antidepressant appears to be working may well be linked to changes it's making in the brain, changes it's making in how we can regulate our sleep and normalize our sleeping patterns. And being able to do that in both a clinical environment where we can do that in our sleep lab, but also doing it in studies where we can actually measure at home as well. It's something which I think is becoming increasingly important and really helps us to understand the importance of sleep in clinical research. Next slide, please. So to very vaguely just finish on the approach that we take to the research that we're doing in MAC in EEG. Clearly takes very dedicated and experienced people. That's actually a slide of me having my EEG measured by some of my colleagues. You need a very dedicated team with extensive EEG experience, and that's exactly what we have. They are proficient in handling a wide variety of EEG devices and systems seamlessly. Because we're a CRO, we have to adapt to what the sponsors and what the clients are looking for on so many of these studies. There's not one size that fits all. Currently, we have studies running which are using things like the NATEX, NeuroWork, SleepWork systems, measuring up to 50 channels of EEG. We have Brain Products 64-channel EEG being used in our later phase facilities. But also we're doing things like the Dream 3 headband, which is a small headband across the head, which is measuring for a limited number of electrodes, but is collecting data both in the clinic and outside of the clinic. It's very important that however we collect this data, and it's analyzed by experts like Professor Doffner and his team at Siesta, these people need to get high quality data. And when you're moving between systems that are being used at all times, that requires a great dedicated and very responsible team. And that's the cornerstone of what we're doing. Not just for simple EEG measurements and QEEG, but we're also doing a lot of work involving things like evoked potentials, auditory and visual. Very important that these can be integrated into protocols in a very standardized and very safe way so that you can get the best data possible and really build that link between the neurophysiological reactions that we're measuring through the brain activity and also performance on various other cognitive tests. The experience of the team is really essential to this. But you're correct there. Next slide, please. So where are we going in the future with it? Let me take you back to my little desk back in my laboratory many, many years ago. The problem we had in the old days was you have a big computer, loads of amplifiers, a few electrodes glued on. It was a nightmare to do. These days, things have become much more streamlined and developments are really moving on at a great pace. This explains to an extent why there is a big gap in information between the early days of EEG research in the 80s and 90s, about a 10 or 15-year gap where people moved across to using these brain imaging fMRI, which proved expensive, time-consuming, and slow. But now that we're getting better systems available, I think it's more that people are moving back to getting EEG into studies earlier to get those early indications of brain activity. And there's a lot of quite exciting things that are happening at the moment. Looking to the future, I think EEG technology holds enormous potential to revolutionize clinical research a lot further in areas such as artificial intelligence. AI-driven analysis of EEG data could lead to discovery of a number of new biomarkers, improving patient stratification, and really helping us predict treatment response with much greater accuracy. EEG is also moving out of the lab. We're no longer reliant on these huge computer systems. I mentioned before the Dream 3 headband we're using in a study. We're able to put a headband on in a clinic and measure in a clinical environment. Or if you want to look at a more normalized setting with people in their home environment when they're taking a medication, when they're with their family, where there are distractions of life, such as television, children, feeding, that sort of thing, Dream Headband can be collecting data. Really, you're not dependent on the setting of it. It's really a fantastic opportunity to measure brain activity in real-world situations, as well as a clinical laboratory. Additionally, I think the integration of EEG with digital phenotyping and other wearable devices holds tremendous promise for a more personalized approach to treatment. Combining EEG data with behavioral and physiological data, we can create a more comprehensive profile of patient conditions, allowing for more tailored and precise intervention. Just to summarize in a conclusion, as far as we see it across all our phases of the work that MAC is involved with at the moment, we see EEG as very much at the forefront of technology, and we're leveraging state-of-the-art equipment to really get data which is helping companies to make decisions at a much more early stage and with much more certainty, which is very important. They're also able to do this without actually really increasing the overall cost of a program. This is often things which is added in. So it's helping us to accelerate the development process, but it's also enhancing safety and supporting innovative research really across a whole new range of therapeutic areas, as we're seeing with the psychedelic research. I think as EEG continues to evolve, we're really excited about the growth in personalized medicine and precision neuroscience. That's something we're committed to really staying at the forefront of. think what I tried to do here is really set the scene then for how we're using EEG in many, many different ways, from early phase right through to the late phase trials. The second part of the webinar, I'm going to hand over to Professor Dorfner to really talk a little bit more in detail about specific use of biomarkers and how they are helping really across a whole range of therapeutic targets. So I'll pass over to you, George.
George, you're on mute.
Thank you, John. Also, a big welcome to all the participants. I would like to now talk a little bit about what we do at the Siesta Group. The Siesta Group is a company, we're a focused service provider or vendor of services specializing in electrophysiological and related biomarkers and endpoints. And in this way, we very nicely complement each other with MAC in our partnership. And the way we do this is highlighted here in the first slide, where we start with our big picture, a holistic look at the EEG field. EEG, as compared to other types of clinical trial endpoints, can be rather complex procedure, and for it to get you the right kind of signal in your trial, it depends on a number of factors, and they are depicted here. On one hand, it's the proper implementation of the procedures and the high quality measurement, and this is where a partnership like ours with MAC really pays off, where we have an experienced site that has done EEG many times before. The second factor is the type of paradigm, both in measurement and analysis that you choose. There are several, as I will be hinting upon a little later. And then, of course, and that's our core expertise, the central analysis. And when it comes to analysis, we do cover the whole spectrum of what modern EEG analysis looks like. On the one hand, classical types of quantitative EEG. We call it feature-based because here is very well defined of how, with the help of signal processing procedures, you arrive at a number of endpoints like frequency band powers. In the standard pharmaco-EEG paradigms, like the quantitative EEG or event-related potentials, but also safety would belong in here. On the other hand, we're, of course, as mentioned before, a company that also focuses a lot on the novel types of applications of artificial intelligence, machine learning analysis. And here we need to distinguish some part of AI is already established and validated here, especially in the area of sleep, where we do sleep staging and sleep scoring. We have fully validated AI-based procedures here, but a lot of other part of AI biomarker of work is more exploratory. So here our expertise comes in, and we help sponsors to find, based on their data in early phase, the optimal type of biomarker based on machine learning approaches, which usually combine many of the features of the left side into a novel complex biomarker, and that can help then in later phases to predict the action of the drug in the later phase. Next slide, please. So here are a few more words about us as a company. So I hinted upon our focus is a little wider than just EEG. What's very important is we're device agnostic, so we're not a manufacturer of any EEG or other device, but we we consult and help find the optimal device for a particular type of study, and the range goes from in-lab EEG, pharmaco-EEG, this is what I'm mainly focusing upon here. We also have a large experience in sleep, just as Dr. Connell mentioned, but that will be content of a later webinar. So today I'm really focusing mostly on the EEG part. We'll say very little about sleep, and also our portfolio covers then more real-world data collecting instruments like wearables, actigraphs, or other types of portable instruments. Next slide. from the very beginning, 23 years ago, we have been a very science-driven company. All of our founding shareholders have been renowned scientists in the field of sleep and EEG. Some of them are mentioned here, like Thomas Pencil. He's our liaison with the American Academy of Sleep Medicine because he was involved in the latest scoring standards. Peter Undera, our chief scientific officer, and other members of our team have been actively participating to standardization procedures in the international societies like the IPEC, the International Pharmaco-EEG Society. We have the core developer of the standard format EDF. We had them him in our founding team. And also, I should mention that our sleep analysis software, Somnolizer, was the first that has received ever the approvals accreditation by the American Academy of Sleep Medicine as automated scoring. Our scientific background is also reflected by a large number of publications. We do work globally. We work with sites all over the world, and we do also collaborate, besides our industrial involvement here, with a number of academic research institutions. Next slide, please. Next. So here now we get a little more in depth of what the EEG procedures actually look like. So there are two main measurement paradigms. In both of those, subjects get patched 19 electrodes, usually with a cap that you have seen in the previous picture. So there's on one hand the resting state EEG paradigm, where the subject is asked to sit either with eyes open or eyes closed in a resting state, not falling asleep, and the EEG is recorded during that the time, and is then later on quantitatively analyzed, mainly with respect to their spectral content and connectivity. The event-related potential paradigm, on the other hand, is one where the subject is asked to fulfill a certain task that involves the presentation of stimuli, either acoustic or visual, and we do, by the way of EEG, measure the responses of the brain to those stimuli, which then reveals a number of important components, like the well-known P300, which is the reflection of cognitive processing of stimuli, which then give us indications about the disorder on one hand and about the effect of the drug on the other. Next slide, please. And when it comes to the analysis, I mean, based on these two measurement paradigms, we can further distinguish paradigms by analysis methodology. So the resting state quantitative EEG, which I mentioned already, would basically look at the full spectral content, then in terms of so-called frequency bands and power in each of those bands, dominant frequencies, and so on, and therefore gives a reflection of the entire brain activity in a resting state. Topography refers to the geometrical localization of the activity, either in a two-dimensional sense along the scalp or even in a three-dimensional. So with methods like the one that's called Loretta, it is possible, based on a larger number of EEG electrodes, to reconstruct the actual 3D localization of a brain activity in the brain, and therefore is in some sense akin to a 3D imaging paradigm that we know, for instance, from fMRI. Event-related potentials require their own way of analysis. Here, there's typically an averaging process over several stimuli to see the average response of the brain to the stimuli. Functional connectivity would take into account a number of electrodes that cover the whole scalp and see how they are synchronized or correlated with each other. And then, as I mentioned, also there is the AI-powered EEG biomarkers. So based on the features that we calculate on one of the other paradigms, machine learning can help to combine them in a linear or nonlinear way to increase the predictive power when it comes to, for instance, response to a certain drug, placebo response, or basically mechanism of action. We can also distinguish by clinical use case. So EEG is a valuable indicator, endpoint, or biomarker for almost every class of psychiatric or neurological disorders. So it ranges from depression, schizophrenia to dementia and other types. I will show you a few examples of some of them a little later. Next slide, please. So here's one example, a very complex type of pattern that you can derive with EEG measurement and the proper data analysis. So what is depicted here is in each of the 4 parts of the image, a number of topography. So every circle represents the scalp viewed from above, and the colors encode positive, red, or negative, blue changes. For instance, drug versus baseline or drug versus placebo. And in this sense, you can, and for the different frequency bands, so you have the multiplicity of depictions here due to analysis in the slower, like delta band, or in the faster, like alpha, beta, or gamma bands. And what you see here is that EEG reflects both what is happening, where is it happening, and when is it happening. What is usually expressed to the type of changes, like increase or decrease in alpha or delta. The where is here a location in the topography in each of those circles, and the when is when you do, as it's depicted here, you do a repeated measurement. You see which effects last long or go away, or which ones are still visible after 24 hours, for instance, or after a long amount of time after dosing. Next slide. Based on these patterns, we can also distinguish very characteristic patterns that belong to certain drug classes. So each column that is predicted here is again topographies of different types of bands in the frequency spectrum, and each column represents one of the classes, like sedatives, antipsychotic, non-sedative antipsychotics, antidepressants, tranquilizers, and so on. And experienced researchers on the EEG side would be able to look at one of those columns and recognize just by the colors and by the patterns what type of drug class this is likely is. And these patterns very often counteract the differences that you observe in the EEG between patients of a certain disorder and matched healthy controls. So one of our founding shareholders, Professor Bernd Saletto, coined this as the key lock principle. So EEG reveals which drug is a key to what is missing or is all right in a patient, the lock, and in order to predict the optimal mechanism of action with respect to treating the disorder. Next slide. Here, this is an example of the 3D localization effects that I have talked about. So the low resolution electrophysiological tomography, LORETA, is a mathematical method to take the EEG surface measurements and reconstruct a pattern of activity in the 3D representation of the brain. So we can locate whether activity, for instance, comes from the temporal lobe or from the occipital regions or for some other parts of the cortex. Next slide, please. So here now, just a few examples very quickly, how really each type of disorder has its own characteristic changes in the EEG patterns, and how we can detect them, and how then we can also predict whether a drug is counteracting those differences. So here's an example of depression. To the left, the frontal alpha asymmetry. So depression is known to increase frontal alpha activity, which can be shown clearly as an effect vis-a-vis placebo. Or there's a positivity in the reward positivity in a event-related potential sense, but also connectivity to the very right is usually known to be increased in a depressive state, which can be seen in the EEG. Next slide. These are very well-known biomarkers about Alzheimer's disease. So on one hand, quantitative EEG, it typically shows a slowing of the spectrum, so increase in the delta band, so in the slow waves, and decrease in the alpha band, so to represent the wakefulness states. Is here depicted on the left, and on the right, it's very well known that the P300 component, the component that you see when the subject reacts to an abnormal stimulus, which the subject is asked to either count or otherwise classify, that this P300 component in Alzheimer's is is delayed, so the latency of the is lengthens, and the amplitude is decreased in early dimension. Next slide. It's an example of psychedelics that Dr. Connell also talked about. So here we generally see an overall broadband drop of EEG power when the psychedelic effect is in place. We also see alpha suppression, and we see, based on the type of psychedelic drug vis-a-vis other types of similar drugs, very distinct connectivity, increased connectivity patterns as an effect of the drug. Next slide. Fragile X and autism spectral disorder also has its characteristics changes. I will skip over that for now. We can come back to that later for the sake of time. And finally, next slide, please. Schizophrenia is also, there's several biomarkers are known, like the mismatch negativity is affected in schizophrenia patients. The EEG synchrony, when you present a click train as a stimulus, is affected, and again a slowed alpha peak. So where the peak of the alpha is located changes position vis-a-vis controls. Next slide. So with these few examples, I would like to wrap up and say, now come back to, again, what we actually do here as a service provider. We guarantee the maximum quality of all those measures, of all those endpoints, and our three pillars of quality are depicted here. So on one hand, it's the site, and here we have, in our collaboration with MAC, that is very well covered with an experienced site that has personnel that has done this many times before. So typically in other trial, we would train and certify the sites to do the procedures properly. Then the workflow management is another important factor here. So we have 100% rule. So we always look at every data set immediately after we receive them. We don't wait until the end of the trial and then look at the data. And then, of course, our way of doing the central analysis with our proprietary algorithms, with or without artificial intelligence, is the third important factor for high quality endpoint and finding the signals in this type of measurement. And next slide, please. With that, I come back to our collaboration that we're depicting here. So MAC and the Siesta Group nicely complemented each other here, combined to fuel the momentum for the sponsors in the trial. So on one hand, a highly experienced site that also has a full service operation and experienced personnel that know how to do proper measurement, and on the other hand, our experience, several decades of working with EEG-based biomarkers, our capability to advise on the right paradigm, and our core capabilities of doing the data analysis, which is based also on our scientific background, and it's state of the art with respect to methodology. In this sense, I would like to wrap up here and hand back to Claire. Thank you very much for your attention.
Thank you so much, George. Thank you so much, John. Absolutely fascinating. I learned a lot personally. I'm sure everyone else did in our audience. So we have some time for questions, which is fantastic. And we have quite a few that are published as well. So I'm going to work my way through those ones. In the meantime, if any of our audience would like to raise their hand and ask a question verbally, you can activate your cameras as well if you'd like to come up on the screen. And I think we'll have our speakers large on the screen, if that's possible, please. So we'll start with the first question that I received. What are the cost-benefit trade-offs of implementing EEG biomarkers in large-scale clinical trials? John or George, would you like to take that one?
Well, George, take that first.
Well, I mean, it of course depends what you compare it with. But if you compare it, for instance, with brain imaging, there's a very clear cost efficiency here. I mean, the procedure of EEG is involves much fewer costs, but also much less burden for the subjects, and is therefore also what the number of repeated measurements is concerned is much more easily implementable, also in an early phase trial.
I'd massively agree with that. I mean, what we're seeing at the moment in some of the studies we're involved with is that about the patient burden, and going into imaging scanners is quite an unpleasant experience. You need to go to a centralized center, whereas with some of the EEG work, because it's much more portable, you can move the experienced staff to the patients themselves and to their nearby clinics. They're going to local clinics, and you can collect really high quality data very quickly. And that seems to be very appealing, I think, but from a patient perspective. And we always need to bear in mind the burden on the patients.
Absolutely, thank you. Next question. What AI validated based procedures are done in sleep analysis, and which ones are hoping to apply in the near future?
I can make a start on that one. In our sleep lab, we can do like a full head of polysomnography where you can have multiple channels of data. It looks fantastic. You're measuring for an 8 or 9 hour time period. It's meaningless unless you can have experts who can actually look at this data and interpret it accurately. There are programs which will break down sleep into various stages, but you still need the overread by people like Siesta, by ourselves, who can give that. Moving away from the full scale, I think we're seeing an increased use of wearables. We're doing some work, as I mentioned, with the Dream Band headband, which is really just a limited number of EEG electrodes, where there's interpolation from those electrodes of what actually is happening elsewhere. And even things like an ActiWatch, where body movement used to relate and to make a estimate of what is actually going on in the various stages of sleep is something which really is revolutionizing how easily we can look at sleep. It's moving away from having to do it in a very controlled sleep lab environment, which is still very important if you're looking for rigor control and early testing of drugs, but to be able to look in a home environment or even a daytime environment. I could be wearing one now, and you could be measuring how this talk is actually affecting my brain.
I think we mentioned, I mean, even though EEG plays a big part in sleep analysis, I think it's worth it. It's a separate story, and we will be happy to do a separate webinar on the sleep aspects. That's why we didn't mention it so much. But in a nutshell, the answer to the question is, I mean, in sleep, on one hand, you have the classical scoring of sleep into sleep stages and sleep architecture, but you can also do the same procedures as I showed you, look at the quantitative changes in stable sleep stages. You can have even a PD/PK profile over the night. So there's many other ways of looking at sleep from an EEG perspective, but then also the portable, there are other types of portable EEG devices as well. So there's a whole range of links. And so I'm looking forward to have a separate webinar on that.
That'd be a good idea. I think we should do that.
Mm-hmm.
Absolutely. Great. And our next question, has it been possible to pick up drug treatment effects with EEG in Alzheimer's, and if so, to what extent have these effects already been validated to be linked to clinical outcomes?
Especially with cognitive decline and slowing of the cognitive decline, there's a bunch of work that really clearly shows the evidence and the proof that with like event-related potentials, but also quantitative EEG, you can detect it early on. I mean, we have published, for instance, in a longitudinal study that we show with the P300 profiles that we could predict the type of cognitive decline of the coming year or two. And we also published with, I think it was with Eli Lilly, a paper on showing how even on a short term you can increase the amplitude and decrease the latency of the P300, so you have positive cognitive effects which counteract those decline. So in a sense, if the drug, for instance, is aiming at slowing down the cognitive decline of Alzheimer's, it's a very clear yes as an example.
Yeah, and I'd just add to that, actually. I think what excites me about the use of EEG in that particular area is that the changes that your relevant to measuring often actually precedes measurable abnormalities through other mediums, such as the PET imaging or even CSF sampling. And the beauty is that these are completely non-invasive. And simple resolution, I think, makes them really very, very useful in Alzheimer's research, definitely.
Excellent, thank you. Can EEG be used to detect adverse CNS effects earlier than behavioral or clinical symptoms?
Yes.
Is the short answer. Okay.
Well, adverse CNS effects like seizures, or I mean EEG is the instrument to also detect seizures or seizure-like episodes. So there's a clear yes on that. Other types of adverse events probably won't show up in the EEG. So again, it depends on what exactly we're talking about here.
Yeah, I mean, it depends what you mean by an adverse event, really, because when you take a
Right.
drug, it affects in the brain. And we've seen in studies where we've been looking, particularly in compounds validly away from CNS areas, mainly in pain. Whereas when we've had EEG included as measures, we're seeing things like changes in alpha activity. And this is often at doses where the patients are reporting nothing at all. So in terms of effects, you can certainly detect them and therefore abnormal effects, I think, in some cases, particularly if you're looking at things like sedative side effects of compounds. I think QEEG would pick it up before people actually start to feel it themselves.
Amazing. Okay, how is QEEG used in sleep studies, particularly detecting abnormalities during NREM and REM sleep? I think we definitely need a separate webinar on this, but
I
briefly.
think we need a long time to talk about that.
Briefly, I mean, that's what I hinted upon just before. You can do QEEG during sleep. Normally, the polysomnography is aiming at the sleep architecture, but it is possible to let subjects sleep with a cap and like the 19 electrodes of the 20 system. And what you would do, you would first do the sleep staging, identify stable periods of like stage 2 as non-REM or stage 3 as deep sleep or REM. So and then average the spectrum over those stable periods. So typically 5 to 10 minutes of such stable periods are taken, and that is then results in similar topographies as I showed before for the resting state paradigm.
Thank you. I think our final question in the chat are the full head polysomnography able to detect neurodegenerative conditions, or is it mainly focused on sleep disorders like narcolepsy?
Well, the all-night polysomnography is mainly aimed at sleep. I mean, not only sleep disorders. I mean, we are, among others, working in a working group of the International Society for CNS Trials and Methodology, where we recognize in the course of writing a review article about it that sleep does play an important central role in very many of the different psychiatric and neurological disorders, and that thus sleep deserves to be at least a secondary endpoint in many drug trials, be it dementia, be it schizophrenia, depression, or so on. So the usual the polysomnography aims at looking at this sleep architecture, and therefore looking at what aspects of the sleep is changed, either due to the disorder or due to the drug. But as just mentioned before, we can do a quantitative look at that as well. You can do PK profiles during the night. So once you wear those electrodes all night, there's a variety of things you can do.
I mean, I'd agree with that. Probably it's confusing because most people, when they're doing a clinical trial, are looking for just sleep architecture. But if you have a really good measure, you have good people who can analyze the appropriate areas, I think it could be used for this area, and it should be being used a lot more than it has been.
Thank you. We do have another couple of questions, but I would like to address the ones that came through prior to the live event today. So are there EEG biomarkers of disease response in Alzheimer's that could be earlier endpoints than current fluid and imaging biomarkers?
I mean,
We've already
generally,
touched on it. Sorry, yeah.
do you want to say it?
Oh, you go ahead with that. That's fine.
I mean, generally, one has to say there are very few, unfortunately, longitudinal study that would really prove whether the signal is earlier. But again, with respect to the mentioned event-related potentials and their capability to predict cognitive decline, there are very clear signs that EEG is an early biomarker. Whether it's earlier than other comparable ones, I'm not in a position to fully confirm, but there is work that really shows that you can early detect in an early time frame the progression of the disease.
Yeah, I mean, I think I agree totally. I think in preclinical early stage Alzheimer's, then there is a role for it, and I think it is very predictive. I think by the time the disease is fully developed, I'm not sure it's going to have more, because there'll be other symptoms that are much more easy to measure.
Thank you. How do EEG biomarkers compare to fMRI and FNIRS markers in terms of predictive validity?
Well, we're big advocates for EEG, so we're going to tell you about how it's got superior temporal resolution, how capturing neural activity is down to milliseconds, which of course is not in the other imaging modalities, how it's much more safe and easy to put people into a comfortable environment with a headband on compared to these big machines that they're in. There's lots of ways I think it is. George, you're going to comment on that?
Yes, fully agree. I mean, if it comes to really exact localization, then probably fMRI and FNIRS have their clear advantages. But clearly, I mean, it's the time resolution and the cost effectiveness that speaks for EEG. And also, I must say, I mean, if you look at it, both fMRI and FNIRS are measuring the blood flow, and are therefore, in some sense, indirect measures of brain activity, whereas the electrophysiological activity is more direct. It's a summary signal, but it's a more direct measurement of really what the brain, where and how the brain is active. And so that, in that sense, you can expect more predictability to some of the important aspects.
Yeah. And if you're looking for something longitudinally where you've got many repeated measures, EEG is so much more simple to do. And I think, it's a direct measure. That's the important thing. It's non-invasive and it's simple.
Thank you. And can we talk a little bit about biomarkers in stroke?
Yeah, I see there's another question to that respect in the chat. So it's also asked about white matter status, like in MS or stroke. I mean, yes. I mean, there's literature that shows clear, distinct patterns in stroke, like asymmetries if it's a localized stroke, or also spectral slowing, increase of delta, decrease of alpha. I mean, here the relationship with the drug treatment is probably a different one. I mean, you wouldn't get. I mean, what you do when you treat stroke is different when you treat a psychiatric disorder. But with respect to what you see in the EEG, yes, there are clear signs of detecting stroke and MS as well.
Yeah, I mean, it's unfortunate that I don't think EEG can be used in any way to predict that a stroke's likely to happen, or I think it would be very useful. What would be interesting to see if any of these measures, these changes, particularly the change in slow wave activity or the coherence between different regions, whether that improves with treatments. And I'm not fully aware of the literature, but I think it's something worth looking at.
So I think so that we can wrap up within the hour, we'll take one last question. But I would like to reassure everyone that all of the questions that have been put today, whether via email or in the chat here, we will respond to. We'll pull together a full Q&A document so that you can reference in the future as well. So final question then. What are the biggest barriers to translating EEG biomarkers from research to clinical trial endpoint?
Great question.
Preclinical research, huh? Is it asking about translation from preclinical research to clinical trials?
Research to clinical trial endpoints.
If I refer to preclinical research, I mean, there's very well-known evidence of how we can translate from rodents or sometimes non-human primates to humans, but in most cases of drug research, it's rodents, rats, and mice. I mean, they have different brains, obviously, but some of the observations in quantitative changes can be translated into what you expect then in the early phase clinical trial. And so it's actually a nice example of how translational research can work here, even though more data along the whole spectrum would be beneficial if we had more studies that really go all the way from preclinical to like phase 2 or phase 3 with electrophysiological data, but the data that does exist points to a nice translatability.
Yeah, I'd agree with that, because we work really extensively in early phase, but also in later phase. We do see a good translation from the animal work. It's been very important for us. Always room for more work. And I think as we are developing and as technologies are getting better, I think that will become stronger over the years ahead.
Absolutely. What a great comment to end on. We've had a question whether the recording will be available. It absolutely will be. We'll be transcribing the questions and providing the answers, and the recording will be sent out to everybody. So thank you ever so much, George. Thank you so much, John, for your time today. Thank you to all of our audience. Thank you to everyone behind the scenes who's made this happen as well. You can see on your screens here the places to go for more information. Do follow us on social media, both the Siesta Group and Mac Clinical Research. It's been a pleasure, and we really hope that we will be hosting soon our second webinar with the Siesta Group, looking at sleep-wake endpoints. If anyone would like any further information on that or a personal invitation once it's arranged, do drop us an email. Thank you.